Welcome. I'm glad that you got up early. You're going to be richly rewarded. Before I start with the introductions, I want to mention a couple of things. We've got our members meeting tomorrow afternoon starting at about 3:30. It'll be preceded by the Norris Award to Randy Reeves for his outstanding work on conservation, guaranteed to be a good seminar. Then we're going to go into the business meeting, and then we're going to have the award ceremony for this meeting. One thing I forgot to mention on Monday was that we're going to have a call for nominations for the officers of the Society from the floor. The board puts together a slate of candidates, but if you think you'd like to serve as a member, as an officer of the Society for Marine Mammalogy, we encourage you to either nominate yourself or talk to somebody and get your nominations in, because it's a great way to contribute back to the Society. I'm going to begin today by introducing the Conservation Merit Award. So this is the highest award granted by our Conservation Committee, and it's unique in that it's not for necessarily ever for a scientific contribution, but it's for real on-the-ground conservation progress. Last year, our last conference, we gave it for the first time to the vaquita team that was developing alternative fishing gear and monitoring the vaquita population, and that award went to scientists as well as fishermen and even the Minister of the Environment of Mexico. So it's a diverse award, and this year it's going to Alexandros Karamanlidis for his amazing work at saving monk seal in the Mediterranean. The former Society President Kit Kovacs will come up now and introduce Alexandros and some of his work. Thank you.
Good morning, ladies and gentlemen. It's my pleasure to stand in this morning for the chair of the Conservation Committee, Barb Taylor. As you've heard earlier, she's part of a big vaquita team that is down trying to save that particular species. But it allows me the opportunity to welcome Alex to this wonderful award. This prestigious award is one of my favorites within our society because it takes specific note of exceptional work done on behalf of conservation. As Jay mentioned, this year's award goes to Alex Karamanlidis and MOM, the Hellenic Society for the Study and Protection of the Mediterranean monk seal. I've known Alex now for about a decade, under the auspices of the IUCN's Pinniped Specialist Group. His research achievements are notable, in part because of their great diversity. Alex is one of the few in this room that works both on terrestrial mammals and aquatic mammals, and does both exceptionally well. The quality of his work is evident in his extensive publication list. But one of the things that has impressed me most about Alex is his we mentality and operational mode. I've rarely heard Alex utter the pronoun I in the 10 years that I've known him. It's always we. And I think that team spirit and the resultant atmosphere of mutual respect and recognition has been a force that's helped the whole med-monk seal research and management community come together to do the amazing things across many borders under very challenging conditions that they've managed to do. Alex is going to tell us about their successes and their ongoing challenges briefly this morning, and more comprehensively later today in the midday conservation sessions. Alex, with much deserved congratulations and very warm regards, the floor is yours.
Thank you, Kit. Thank you, everybody, for coming out here so early and sharing this moment with me. I really appreciate it. As you can imagine, this is a really special moment for the Hellenic Society for the Study and Protection of the Mediterranean monk seal, but also for Greece, a small country that does not have the biggest of traditions in Mediterranean monk seal conservation and research. It is actually ironic that I should be standing up here and receive an award on the research of the Mediterranean monk seal. I can vividly remember 15 years ago when I tried to set up a PhD on monk seals that I was turned down because, and I quote, "Studying Mediterranean monk seals is academic suicide." In all fairness, nobody could have blamed anybody back then when I tried to do that for thinking that way. After all, up until the year 2000, the news for Mediterranean monk seals was not overwhelming. In the last half of the previous century, the conservation community had witnessed the disappearance of the species from the Black Sea, the entire western half of the Mediterranean Sea, and population numbers in Madeira had fallen to perilously 8 individuals. To make things even worse, in 1997, we witnessed the mass die-off of Mediterranean monk seals in Cabo Blanco in Mauritania, while at the same time, countless symposia and conferences, perhaps as many as monk seals remaining, tried but did not succeed in turning the tide for the species. The only shimmer of hope back then was the establishment of grassroots organizations for the conservation of the species, organizations that had dedicated and passionate people who understood that the future of the Mediterranean monk seal and the battle to save the species was not going to be won in any conference hall, but out in the field, in the schools, and in the hearts of the school children, and on the fishing boats, sometimes perhaps drinking a little bit too much ouzo with a fisherman. Conservation actions nowadays in Madeira are led by our colleagues from the Parque Natural de Madeira Service, and in the landmine-riddled Cabo Blanco Peninsula by Fundación CBD Habitat. And last but not least, research and conservation efforts in the Eastern Mediterranean are spearheaded now by the Hellenic Society for the Study and Protection of the Mediterranean monk seal, aka also as MOM. MOM is a non-governmental organization that was founded in 1988 in Athens, in Greece. We are a relatively small conservation organization that is working in a country that has one of the highest coastline to countryside ratios, a Byzantine-like state bureaucracy that can make any conservation effort a nightmare, and we're trying to literally study a species that must be one of the most elusive of marine mammals on Earth. Therefore, the fact that MOM has managed to play a significant role in the success story of turning the tide for the Mediterranean monk seal, which is honored by this award, makes us both proud and confident to keep on working for this cause. I will not go into further details on how we are going to achieve this. I would just warmly and kindly welcome you to our conservation highlight session later on. We're together with Dr. Littnen from the Hawaiian monk seal research team. We will talk a little bit more about that. I would just like to say, of course, that we did not do this on our own. So on behalf of MOM, I would like to thank the European Commission and especially the Life Funding Mechanism for supporting monk seal conservation actions all over Europe. I would like to thank IFA for being there when MOM was being set up, and the Thalassa Foundation for supporting us now during the difficult times of the financial crisis in Greece. I would like to thank also our colleagues and friends from the Hawaiian monk seal recovery team, with whom we share our common passion for monk seals. Thank you guys, you've been a really big inspiration for us over all these years. The Marine Mammal Commission for supporting us in the last years at some of our more innovative research efforts. The spirit of open-mindedness and cooperation that this organization has shown towards us is unparalleled, and I think you guys in the US can call yourselves lucky to have them. And of course the IUCN Pinniped Specialist List, and especially Dr. Kit Kovacs, who gave the Mediterranean monk seal people a powerful voice to the conservation community. On a more personal note, I would like to thank my colleagues at MOM, Stella Thamantopoulou, Eleni Tunda, and Panos Dendrinos for letting me be a part of the family of MOM. I am sure that that has not been a very easy choice for them, after all. When I arrived at their doorstep, I was a 25-year-old cocky and arrogant biologist with long hair and military boots, so not very trustworthy material. I would have not managed to succeed any of this without the love and support of my family, and especially my wife, who has kept us all together over all these countless days and hours when I was out in the field. Mom would like to dedicate this award to the Mediterranean monk seal, the toughest seal out there you will find, a species that is both inspiring, amazing, and challenging. And we promise to keep on fighting out there for it. We will never surrender. Thank you.
Thank you very much, Alex. And so, yes, please do go to the Conservation Highlights service session at lunchtime to hear more about his work. Okay, so I'm feeling very important, so I've put my hat back on. And it's one of those tricky concepts of the meaning of life that Monty Python talked about. People just don't wear enough hats. So this morning, it is my honor to introduce the Wood Award. This is given in honor of Forrest G. Wood, who was a founder of the Society for Marine Mammalogy and noted for his editorial skills. The award is for excellence in scientific writing and given to the best manuscript accepted for publication in marine mammal science. The winner is selected from manuscripts submitted to the journal during the interval between these conferences. So I'd like you to give an applaud to this year's winner, which is Julie Vanderhoop. Julie completed her bachelor's in marine biology and oceanography here at Dalhousie University in Halifax with her undergraduate thesis on vessel strike probabilities in North Atlantic right whales. She completed her PhD at the Massachusetts Institute of Technology and the Woods Hole Institute with her thesis on the assessment, behavioral, and fine-scale movement responses to added drag. And she is currently a Marie Curie Research Fellow at Aarhus University in Denmark. I actually had the honor of teaching Julie here at Dalhousie many years ago, and it was very clear to me then that this was a student that was brilliant, very driven to excel, and this is quite clear that she has done so. So this morning, she's going to present her winning presentation, Drag from publication, Drag from Fishing Gear Entangling North Atlantic Right Whales.
Thank you, Damien, and it is pretty cool to be back here in Halifax. So first, I really want to acknowledge my co-authors here, both on the original paper that we published in Marine Mammal Science and on much of the work that actually stemmed from this publication. And I'll present a combination of that today. So this is a North Atlantic right whale, and at the beginning, when I was preparing this talk, I thought I would really have to introduce the species and how it's critically endangered, but how we also know that the population has begun to decline. And fortunately enough, we've heard a lot about this week at the consortium. So the current estimates of the population in 2016 is 451 individuals. And entanglement in fishing gear contributes unsustainable mortality to the species, accounting for 36% of the diagnosed mortalities from 1970 through 2009. But entanglement also largely affects the living population. So on the top panel, we see a dead North Atlantic right whale with gear in its baleen, but also individuals that are either carrying gear or that show scars from having interacted in gear. So this is the North Atlantic right whale population, all 451 of them. 83% of the population has been entangled at least once. 26% of the population obtain new scars every year. And today, 16 individuals are currently carrying gear right now. The population spans from here we are in Halifax, down to Florida and back up north to the Gulf of St. Lawrence and Newfoundland. And in green, that's the movement of one animal from sightings data, so it's flying over North Carolina. But these animals migrate to and from often southern and northern habitats, and they continue to do so when entangled, which will happen now when the animal's red. So it's the same individual, and they often will tow gear as they swim through their range. And over the course of months, we often see in these prolonged entanglement cases really significant losses in body condition. So this is the same animal, e.g. 3911, when she was last seen gear-free in February and first sighted entangled in Florida 10 months later. And there's this huge change in the blubber lipid stores of that animal. So this really prompted us to ask, how much energy does entanglement cost? And we began to address this question for this one individual, e.g. 3911, in an earlier paper that we published in 2013. The premise is that typically, when a whale swims, it has to counter drag forces. And when a whale is just a whale, that drag often comes from its body shape and size, also from interaction with the surface and propulsion. But when a whale is entangled, there's a lot more drag involved. That drag can come from the gear itself, but also based on the configuration on the animal. So we have this interference drag that occurs depending on where the gear is attached, how often, and how big it is. So with that added drag, the animal has to counter that, and they do so by producing more thrust, which over time, requires more power, and that leads to greater energy consumption. So the first step in understanding the sublethal effects of entanglement in fishing gear was to measure just how much drag there was on the gear that entangled right whales. So our 2015 study, published in Marine Mammal Science, measured drag forces on fishing gear that had been removed from right whales. It evaluated the drag characteristics of that gear, put those measurements in the context of the animals, and also began to quantify the benefits of disentanglement. The drag measurements and other aspects of this study, however, were really taken further from that initial publication. We've been able to further quantify the benefits of partial disentanglement, not only in a drag perspective, but also in terms of energy and in animal survival. Using biologging tags, we've been able to look at the changes in diving behavior and biomechanics associated with towing fishing gear. We've also quantified the energy expenditure and some of the survival probability based on those measurements of the 15 sets of gear. And also, we've begun to be able to estimate drag on entangled right whales when they're first seen entangled. So today, I'll tell the story of how this 2015 paper came to be, and I'll focus on these last two aspects of the work to talk about how much energy entanglement costs. And I'll also talk about how we can estimate gear on other animal cases, especially in the context of recent entanglements. So in an earlier study, as I mentioned, we had obtained fishing gear that had been removed from that chronically entangled whale that I showed before, e.g. 3911. So this is a diagram of the whale and the gear on its body. That's the gear that we removed. And we towed it on this tensiometer, or a load cell, behind a vessel at different speeds to measure the amount of force on that rope. And this was really great to do for one animal, but it was clear to us that it could be taken further. The North Atlantic right whale is a really well-cataloged species, and all of the gear that's removed by trained disentanglement teams or at necropsy is stored in a gear warehouse by NOAA Fisheries. So we looked through the North Atlantic right whale catalog and through the gear warehouse and really started to design our 2015 study. So we obtained fishing gear that was removed from 15 North Atlantic right whales, and we sought cases where we knew the sightings history of those animals, their sex, their reproductive history, and the duration of their entanglement, so we could really try to look at the drag burden over time and put it in context with those life history events. And so these are these really nice case histories that are put together by the team at the New England Aquarium. And these are 9 of the 15 sets of fishing gear that we towed. And what I want you to take away from this is just how different those sets are. So some of them are short pieces of line, other are much larger, much longer, or have sets of buoys attached to them, or lobster traps. And we were able to really quantify the differences in drag characteristics on those sets of gear by towing it off the Hussey RV Tioga. So this is myself and David Morin in 2012, about to deploy a lobster pot attached to this tensiometer off the back, and then also another set of gear that was much longer with a buoy on the end as it's being towed. And we towed these at different depths, but also at a greater range of speeds than that initial 2013 analysis. So what I'm showing here is the drag force measured on only 5 of those sets of fishing gear, just to simplify things a little bit. And this is just to show that the gear that we measure on the drag, or the drag that we measure on the gear, really depends on the configuration. So there are some of these sets of fishing gear at the bottom here that are much smaller, and they don't add very much drag. And there are some intermediate cases here that are very similar in their drag magnitude, but the sets are quite different. So one of them is much longer than the other, one of them has buoys. And then the heaviest gear set that we towed was 35-pound lobster trap, and that, unsurprisingly, had the largest drag forces that we measured. But it's important to put this in the context of what a whale would experience normally. So here in the orange is the mean and then the minimum and maximum drag that we had towed on those 15 sets of fishing gear. And if we can estimate the theoretical drag on a whale's body, which I'm showing here in blue, we can also, based on mostly airplane physics, actually estimate the interference drag. So that's the drag that comes at the point of the attachment based on where the gear is on a whale's body, and that's in purple. So combining all of those leads to the total drag forces on an entangled whale's body. So at minimum, drag increases, or gear increases drag only negligibly. On average, drag from fishing gear will, for the cases that we towed, will increase drag by 1.5 times. And for the heaviest set that we towed, that 35-pound lobster trap, it increased drag by 3 times. Now going back to that animation at the beginning, we see that these animals tow gear for a long time throughout their range. And that means that they must overcome the drag and continue to move. And we wanted to know then how much energy it takes to do so. Oh, and sorry, this is just looking at 1.3 meters per second, a cross-section of this plot to see kind of the mean and max contributions of drag. So to ask our question of how much energy does entanglement cost, we needed to come at it from 2 ways. One was looking at the more physical side, so the drag, the power, and the work required. So this is the thrust required to swim when not entangled in black or when entangled in blue for those 15 animals. And the second was based on the consumption of lipid energy reserves. So this is a North Atlantic right whale that was in great body condition. It died of a vessel strike. And by comparing the changes in the measured blubber thicknesses or estimated blubber volumes between a normal right whale and those that died from entanglement, we can see the changes in energy consumption. So if animals don't slow down, entanglement increases the power output. So here I'm showing the, I'll just mention that it's at this point here that we kind of began to stray away from that 2015 study that we published. So this is now taking some of those measurements and applying them more broadly to the population. So this is the thrust for animals when they're not entangled and when they are entangled in their respective gear configurations. And the red ones are those that died, and the black ones are those that survived. And here is the difference in thrust required to move. So this is the difference in the amount of thrust required for me to overcome the drag from my specific entanglement. And that's really what matters. And that additional thrust, or gear increased thrust by around 1.6 times. But the amount of extra power, this difference, over the duration of the entanglement is really what matters. So that is the amount of work required. And we can estimate from that entangled whales, or these entangled whales, expended somewhere between 4 to 40 gigajoules of energy just to overcome the drag in their specific entanglements. From the other angle, we compared measured blubber thicknesses from necropsies of dead juvenile and adult right whales that died from entanglement or that died from other causes, that were in good body condition. So these are the dorsal blubber thicknesses here. And juvenile and adult right whales who died from entanglement have 50 and 17% thinner blubber layers. And by comparing those thicknesses and integrating over the whale's body, we can estimate the blubber volume, and that blubber volume consumption over time is somewhere between 70 and 100 gigajoules of energy. And we see similar losses in body condition and blubber thickness over time from pregnant right whales to lactating right whales. And that's the most energetically costly life stage in these species and in marine mammals in general. So that really begins to put in context the energetic cost of entanglement. So from these two methods, one from energy output, from the drag and thrust, we can get an idea of how much more energy it takes to swim when entangled. But from that energy input from blubber consumption, we can also get a better understanding of how much energy these animals are consuming, and then putting that in the context of the life history events. And the blubber number is also slightly higher because it's a bit more holistic, and it's also over the course to death. What about other life history events? Migration has been estimated to be about 15 gigajoules, and calves have been estimated by the same method, actually, to cost 580 gigajoules. So here I'm showing the energetic cost per day of different life history events from the literature, so foraging, migration, pregnancy, and lactation. And the blue bars are the energetic cost per day of the entanglements for those 15 cases that we measured. So per day, the costs are comparable to foraging, migration, and pregnancy, and on a per day basis are somewhere between 3 to 34% of the daily costs of lactation. Based on copepod densities and published assimilation efficiencies, this equates to about 1 to 2 hours of feeding on their most productive foraging grounds. And entanglements can also last as long as these life history events as well. So here I'm showing the minimum in gray and maximum in white entanglement durations of 72 North Atlantic right whales. And this work is put together so beautifully by the New England Aquarium and the Right Whale Catalog. And so from this we can see that many or almost all of these entanglements last longer than it takes to migrate. Many of them last longer than it takes to produce a calf and wean it. And some also last just as long and longer than the historic calving interval for the species. So entanglement, therefore, has an energetic cost and duration that's comparable to reproduction. And we wanted to put this in the context of the energy required for a female to produce a calf, and to see how long it would require to recover from that energy burden. So here I'm showing the relative costs for a reproductive female, and this is really based and is comparable to some of the work by Stella Villegas-Altman and Dan Costa, where we have the proportion of the 4-year energy budget. A large proportion of it goes to maintenance, some to lactation, and minimal amounts of pregnancy. And then these are the energy burdens, or the minimum and maximum durations of the entanglements for the females whose gear we were able to measure. And so entanglements require about 0 to 8% of the 4-year female energy budget for a reproductive female. And that takes somewhere between 1 to 3 months to recover from, but also up to years. And again, these were only the gear that we were able to tow for those females. Now, at the same time, we've seen very variable calf production and an increase in the mean calving interval of reproductive females. This jives really well with some of the work by Amy Knowlton, where here I'm showing the proportion of females in the North Atlantic right whale population. The median calving interval is around 3.5 or 4 years, but there's also a large proportion of the population here that takes much longer to calve. And in the most extreme cases, these are the females that are presumed dead, so that have had a calf and we don't see again. But there are also females with severe entanglements that reproduce less frequently than females who are not entangled or without severe injuries. And Amy's also shown that those that are carrying gear or with severe entanglements are less likely to calve again. So this work has really come together to show how entanglement from these 15 whales actually, whose gear we towed, can cost as much as migration, pregnancy, and foraging, that it can last as long as the calving interval, and that it can take 1 to 3 months or up to years to recover from a female energy budget perspective. Now, considering the population trajectory that I'd showed at the beginning and the proportion of the population that is affected by fishing gear, it's important to know that we only towed gear for 15 whales. So could we have learned something from that initial study to apply to free-swimming whales right now? So from the gear sets that we towed, we were able to look at their dimensions and establish relationships between the dry weight of the gear on top or the length of the gear on the bottom with the drag forces that we measured on the boat that day. So now we can predict the forces of gear on whales at the time that they're first sighted entangled if the gear is described. So for example, if a whale is reported to be towing 125 meters of line with floats, it's estimated to have around 150 newtons of drag. In this way, we can estimate the drag on a whale when it's swimming. That can be used to look at the severity of the drag burden in terms of the serious injury assessments that are done by NOAA Fisheries. And we've also been receiving some recent inquiries about a specific type of gear that's becoming more frequently observed. So there are many recent entanglements in snow crab gear in fin whales, humpback whales, and also in North Atlantic right whales, and this is a snow crab pot here. We've had 5 live entanglements in snow crab gear this summer in the Gulf of St. Lawrence. 2 of the whales in the Gulf of St. Lawrence that died this summer had interacted with snow crab gear. One case is starboard that I'll talk about in a moment. And another right whale, Ruffian, was last seen gear-free up in Anticosti, and he was disentangled here in Florida carrying this lobster, or this snow crab pot, sorry. And when I was asked to kind of put these numbers in context and to maybe estimate the drag forces on that gear, I realized that it was pretty far beyond what we had measured before. So I mentioned that we towed a 35-pound lobster pot, and that's 15 kilos. But this snow crab gear is much larger. So the gear on Ruffian, so I'll walk through these 2 cases, Ruffian and starboard, and estimate the drag on those animals. So the gear on Ruffian was so beautifully laid out and documented here by the disentanglement team in Florida and Georgia. And it's 138 meters of rope and around 19 kilos of line. So from that, we can estimate that the drag from the line is somewhere between 80 and 170 newtons. So on this side here, I'm showing the drag on the body and then the drag on the line. And in these 2 estimates are kind of minimum and maximum bounds because there's a bit of uncertainty there. And then if we begin to consider the pot, it's a lot larger. So these pots are 180, or this pot was 181 centimeters, and that's how tall I am. And it weighed 71 kilograms, which is how much I weigh. So it's a lot larger than a 35-pound lobster pot. But still, using the best estimates from the lobster gear that we had measured, we can estimate that the drag was somewhere between 350 and 900 newtons. And that's for an empty pot. And it also doesn't consider that this animal was likely towing it along the bottom because the length of the gear was much longer than the depth of the water column, at least in the water that it was disentangled in. Now, as for starboard, this whale was entangled in only 30 meters of line, and so that added only around 70 newtons of drag. But it was wrapped around the body quite a few times, and it involved both pectoral fins and also had some floats attached. So here we can see the drag on the body. The interference drag is quite high, and then we have drag on the line. And the y-axis here is actually double what it was last time because this animal was entangled in 2 pots for around 4 days and up to 4 pots for between 1 to 5 days. So each trap weighs about 100 kilograms, and the drag in this scenario is pretty much more than I could have considered. So this is the same body drag in black, interference drag in gray, and gear drag in white that we had published in this paper in Marine Mammal Science in 2015. In this case here was the 35-pound lobster trap. And then here I'm showing those drag estimates for the two animals entangled in snow crab gear with the same color scheme. So we can see that some of this gear is quite different. Other aspects are similar, so the amount of line and the weight, and that allows us to make some of these estimates. But it's pretty important for me to make some of the estimates on the traps fairly conservatively. Now, the Canadian snow crab fishery was closed towards the end of its season due to increasing entanglement rates and also high right whale mortality in the area. And I'm not sure what the plans are for next year, but it is pretty important that we recognize that this threat is fairly significant from a sublethal energy perspective. Now, the drag is quite high, but it's clear that these animals can tow them thousands of kilometers, as was shown by Ruffian. But for other cases like starboard, it can be so high as to anchor animals in place for the death of starboard. So our 2015 study set out to measure drag on fishing gear that we had removed from entangled right whales. And in doing so, we were able to quantify and also put in context the drag forces on gear for those animals. These measurements have since been combined with modeling, biologging tags, morphometrics, necropsy data, and other gear types to get a more holistic understanding of the sublethal and population effects of entanglement. Entanglement is a leading cause of death, but it's also sublethally exceptionally costly and can last years, effectively removing individuals from a reproductive population. So it's important, I think, to begin to consider entanglement as its own life history stage. And I would also recommend that we focus some efforts on really understanding the risk of these other gear types. With that, I'd really like to thank everyone involved in this study and all of the work stemming from it, as well as, all of the people who've really come to get a better understanding, again, of this holistic understanding of the sublethal impacts of this threat. So I'd like to thank our funding sources here, the North Atlantic Right Whale Consortium, the Hui RV Tioga, and especially my PhD advisor, Michael Moore.
Thank you, Julie. So we have lots of time for questions, and there are microphones set up. So if you'd like to pose your question from them, that'd be great.
Hi. Now, in talking about what you did, you basically just concentrated on the drag, but you didn't talk about performance, which can affect things. So one was the animals towing the fishing gear have a lower speed, and then is there any indication of how this may affect maneuverability, which will affect their foraging ability?
That's a great question, Frank. So a few things. One is that was one of our really big first questions was, do they slow down? And so we looked at some of the data from the telemetry buoy and some other tagged right whales from Mark Baumgartner, and we couldn't see a really big difference in speed, but that's also because of the resolution. What we did see in our recent paper on the biomechanics looking from DTAGs is that we see significantly slower descent speeds and ascent speeds. So those are still, those do have to be taken with a grain of salt because they are based on the pitch and the descent of the animals, but we did see significantly slower speeds when they're entangled compared to after disentanglement.
What about depth? Dive depth, was that adversely affected?
So we have 2 times now that we've deployed D-tags on entangled whales. The first one was this beautiful, beautiful plot where you can see without even knowing where the gear was removed, that there's a huge decrease in the dive depth and the dive duration. The second case was less clear. So what actually happened in that animal is that they deployed the D-tag, they removed some gear, and then they attached the telemetry buoy. And so in doing so, they actually added a bit of drag for a short period of time. And we didn't see as clear of a diving signature in that animal. So it's interesting to make inference from n equals 2. We know that they do different things.
What kind of data are available on re-entanglement by individuals?
On which, sorry?
Re-entanglement?
Re-entanglement. So Amy Knowlton and the group at the New England Aquarium have done a great job of saying, or of looking at how often animals will get entangled. Many get entangled, 83% have been entangled at least once, as I mentioned. Many become entangled more times, and some whales have been entangled over 4 and up to 7 times.
I got that impression from you. So my question is, would you dare to speculate on whether there's any learning involved in avoiding the problem?
It's a great question. I mean, we know that some do get entangled every year. I don't know how the severity of those interactions change over time. I'm not sure if Amy would know either. But we at least see scarring repeating on those animals. So it's very hard to know.
I know, it's a hard question. And one other one is probably hard too, but can you just comment briefly on the pattern of entanglement? In other words, would it imply some sort of basic behavioral response, a twisting or something?
I think we all wish that were the case. Entanglements can be so different, and there are some that just involve one line that goes through the baleen, and that's a great case because those animals will often shed that gear on their own. Other entanglements, the photo that I showed of starboard, for example, that was an extremely complex case, so there are multiple times that it will wrap across the flippers and around the body. So we don't quite know how these animals are interacting with the gear and what leads to that. There have been really great efforts, for example, at Duke involving a simulator to try to re-engineer or backwards engineer some of the configurations that we see to get a better understanding of how it actually happens.
Julie, that was a stunning presentation. I just want to tell you that. Yeah. One of the other issues, of course, is that sometimes the entanglement directly interferes with an animal's ability to gain energy intake. And I just wondered if you could talk a little bit about that in any way that you wish to.
Yeah, so the blubber thickness measurements that we did were kind of to see if we could get a better understanding of all of those effects of drag. So you not only have maybe increased energy output, but you may also have a limitation to feed. So understanding a bit more of how that gear interacts in the mouth, how a reduced energy intake can occur by that way. And it also depends on what time of year this is happening, right? If an animal becomes entangled when it's fasting, it may be in a much worse scenario than if it gets entangled on a feeding ground, even if its baleen is not impacted. So understanding kind of how the mouth entanglements affect feeding is still a good focus for research.
If a right whale gets entangled in snow crab gear, when it dives down, how much harder is it for it to come back up?
That's a really, really good question. And it depends, I guess, on how buoyant the rope is. Going down might actually be easier because the gear's already down there compared to going up. So looking more at kind of the modeling perspective of that would be really interesting, or continuing maybe to tag animals when we see them entangled. Thanks, Damien.
Okay, let's thank Julie again, so I've got ice in my mouth. Here you go. Okay, well, we're doing very well for time, but might as well press on. Okay, so it is my delight to be able to introduce our third plenary speaker of this conference. So again, we have two contrasting plenary speakers that will provide insights. One who is in the mid-career and another one who is well established. That's got nothing to do with age, by the way. And they have very different approaches to their work, which you will come to understand. There's also a very stark contrast in the amount of head hair between these two speakers, which you will also get to appreciate shortly. So the first speaker, Hal Whitehead. Well, many people know Hal, but I will introduce him nonetheless. He is a professor here at the Dalhousie University in biology, and he has a background in mathematics and zoology, which he has very wisely cemented together. His research focuses on social organization and cultural transmission in the deep water whales and on their ecology, population biology, and conservation. Hal's approach is one that is very much unintrusive. He is one of the few biologists that collects data using direct behavioral and acoustic methods from his 12-meter sailboat. So I want to just bring something that you don't know about Hal, some insight into this man. So I thought I would ask past and current students in his laboratory to provide me with one word to describe him. Salty. Curious. Devoted. Intelligent. A real gentleman. Jovial. Complex, like his stats models. Humble. Generous. Bubbly. Kind. A soy sauce lover, and a heart as big as his hair. So this morning, Hal is going to present on the gene culture evolution in whales and dolphins.
Thank you very much, Damien, and thank you all for being here. I have a very hard act to follow. We've heard two exceptional conservationists, and I'm not going to talk about conservation. But conservation is vital. And luckily, I was given something to read out. So this is my conservation pitch. There are alarming efforts in the US Congress to undermine the Marine Mammal Protection Act, and here there are some NGOs circulating a statement against this, and I hope you'd all consider signing it, and if you do want to sign it, go along to the Oceana NRDC booth. Right, now away from conservation. I'm going to talk about an idea which has intrigued me for about 20 years, and that's recently come back into focus, propelled by new results and some new ideas. So I'm going to discuss some, what I think are pretty far-reaching ideas, but the proof is usually uncertain. And there's certainly scientists out there who are pretty skeptical about some of them, and there's probably some scientists in here who are also pretty skeptical about some of them. Right, so first, I've depended on this with help from a number of people, and the first person is Luke Rendell, who has propelled my ideas of culture, and we've worked together on a lot of this. And I'd also like to thank his colleagues in Britain who are also working with us. But I've needed even more help on the genetic side of this, because that's not my background. So I'm very grateful to those geneticists who've helped me try and understand things. And finally, through this talk, one species is going to keep coming up. And it's not a species I study, except when they try and eat my guys. And so thanks to the killer whale people too. So I'm going to talk about culture. Culture, as Luke and I define it, and in fact most zoologists who think about it defining, is something like information or behavior acquired by social learning and shared by members of a group. So the two key bits of this are first the social learning, that's how the information is moving around, and secondly, that it's shared, it's a communal thing. Now there's a whole taxonomy of culture, but, which I'm not going to go into, but I'm just going to mention one dichotomy, and that's between vertical cultures, where the social learning is from parent to offspring, and horizontal learning, horizontal cultures, where the social learning is between peers. So if we take that definition of culture, it's a system of heritability. So information is moving from organism to organism, so their phenotypes are like one another. So it's an alternative transmission mechanism to genes. So I like to think of a population with these two streams of information running through it. The stream of genetic information, the stream of cultural information. Now these two streams can interact. You need genes to produce brains, sensory systems, and other things which allow culture to work. And I'm not going to talk about that. I'm going to talk about the other way, where the cultural stream may be affecting the genetic stream. And I'm going to talk about it in reference to the whales and dolphins. And the reason for doing that is not just that many of you are interested in these creatures, but that there's remarkable evidence that culture is an important aspect of their lives, given how hard it is for us to study their behavior. So to pick a couple of highlights, for killer whales and sperm whales, they have stable, sympatric, multifaceted, in other words, different kinds of behavior cultures. In the bottlenose dolphin, we have ample evidence from the field for behavioral variation, some of which we know is socially learned, often vertically learned. But we've also got, from captive studies, really good information which suggests that these are perhaps the best non-human imitators there are. Cetacean cultures include both horizontal cultures, so things like humpback whale song, and that time when all the southern residents, killer whales, decided to spend the summer pushing a dead salmon around, and then it wasn't cool a few months later, as well as vertical stable cultures. So the killer and sperm whale dialects, the bottlenose dolphin foraging strategies. So Luke and I conclude that culture is an important determinant of behavior, and we would say for many whales and dolphins, and when I'm feeling a bit punchy, I'll say for most whales and dolphins, and after a few drinks, I might say for all whales and dolphins. So I'm going to talk about 5 ways in which that cultural stream may affect the genetic stream. The first is through plasticity. The second is to do with speciation. The third is to do with genetic diversity and cultural hitchhiking. And the fourth is kind of the standard ones, the evolution of functional genes in different parts of a species. And finally, the evolution of functional genes species-wide. So here's a pretty plastic dolphin. And the idea here is that horizontal cultures allow adaptations to new environments. So if the environment changes in a place, or the animals move to a new place and find a different environment, they can figure out how to deal with it, not by evolving new genes, but by by changing their behavior, by learning from each other. And a possible example of this is here. This shows all the odontocete, well, not all, but many of the odontocete species and their latitudinal range. So on the right side, the species which are found nearly from pole to pole, and on the left side, those which are just found in small parts of the latitudinal range. And if you wonder which species these are, the three I've mentioned, because they have good evidence of culture, are the three at the right. So the idea here is that culture initially buffers genetic selection, so reducing genetic selection, and it can lead to the use of new niches, and that can subsequently, by something that's called the Baldwin effect, lead to subsequent genetic evolution. The second form of gene culture coevolution I'm going to talk about is speciation or radiation. And I'm going to do this in relation to the killer whales. So as most of you probably know, killer whales eat all kinds of stuff, from herring to blue whales. But each killer whale usually has a very much more reduced diet than that. In fact, they're extreme specialists, or most of them are. So here I illustrate the different kinds of food that are specialized on by the best known ecotypes in both the northern and southern hemispheres. So this is pretty extraordinary. We've got this radiation into these ecotypes, which Phil Moran and his colleagues have suggested should be separate species or maybe separate subspecies. So how did this happen? Well, the killer whale scientists seem to have converged around a scenario which has been best, I think, expressed by Rüdiger Reich and his colleagues. And the first thing that happens, and this happens in other species too, like sperm whales and bottlenose dolphins, is that cultural feeding specializations of the matrilines divide the populations. And then, this is the key part, to use their words, cultural differences have resulted in sufficient reproductive isolation, even in sympatry, to lead to incipient speciation. And they suggest three classes of ways this could happen. First, cultural xenophobia. So if you're a member of a particular ecotype, you really just don't like the other ecotypes. Second, the cultural specializations prevent dispersal between ecotypes. So if you're brought up feeding on Chinook, it would be pretty hard to start feeding on harbor seals. And third, these cultural divergences may have led to post-zygotic differences, barriers, through either neutral processes, drift, or through gene culture coevolution, and I'm going to talk a little bit about that later on. A puzzle here, though, is if you look at Phil Moran's picture of the evolution of the different ecotypes, you can see they go back at the most a few 100,000 years, whereas killer whales evolved maybe 8 million years ago. So if you've got this wonderfully efficient method of producing new species, subspecies, why don't we get divergence much further back? Well, I've been thinking about that, and this is the scenario that seems to make a bit of sense. That you've got this culturally driven divergence into ecotypes. The ecotypes become increasingly specialized. The populations are small, and then they're vulnerable to ecological change. which can, because they're specialized, small populations, they're very vulnerable, and maybe they even destroy their own resource space. And so the extirpations are common, and you get the ecotypes blinking on and off. So with some help from John Ford, I've modeled this, and I'm showing this here. So if you think of the x-axis in this diagram as the resource space for killer whales, so herring are on the left, perhaps, and blue whales on the right, and there are different things they can eat in that range with different abundances. So we're going to start off with a bunch of ecotypes which are generalists, and that's shown at the top by these broad curves. And they're going to be yellow. And as they move along, some of them may decide to specialize, and they're going to become bluer. So here we go. Here are the macrolines in yellow. Now we're getting a bit of specialization. The niches are narrowing a bit. They're focusing on the areas where there's food in that area. They're becoming increasingly specialized. Now, if you look at the one on nearly on the far right, it's focusing on a very tight food source below. It's specializing. The amount of food there is going to zero. That bunch of killer whales has been extirpated. So at the end of all this time, we end up with just 5 ecotypes left. If we run the same starting conditions, everything the same except no specialization, this is what you get. So this is the remarkable scenario that seems to be happening with those extraordinary animals, the orcas. The next form of gene culture coevolution I'm going to talk about is cultural hitchhiking. This is when culture reduces the diversity of genes which are being transmitted in parallel with selective and heritable cultural traits. Now that's a bit of a mouthful, so I'm going to try and show you how it works. Now in this case, say imagine this is an ocean with a bunch of sperm whale groups, or in fact clans would be more appropriate. The size of each of those pies relates to how many animals are in that group. And the colors in the pie indicate the mitochondrial haplotypes. So quite a few of them just have one mitochondrial haplotype, which is consistent with what we know of sperm whales. Some of them have 2 or occasionally 3. Some are bigger, more animals, some are smaller with fewer. And that's what happens after running an agent-based model for a few 100,000 generations. Now, if you also notice that there's some red crosses, and this is the cultural part of their lives. At the moment, all those red crosses are the same size. But as we run along, I'm going to allow different groups to get good ideas. Cross gets a little bit bigger. Their reproductive rate goes up very slightly. Some will get bad ideas. Their cross gets a little smaller, and the reproductive rate goes down. And at the top here, we'll keep track of the genetic diversity. So here we go, and things are increasing and decreasing, but the light blues have a really good idea. I don't know what it is, it's not nuclear weapons. And they're taking over the ocean, and genetic diversity has crashed. So on the left here, we've got the scenario which goes along after hundreds of thousands of generations, and suddenly we just give little tweaks to the cultural behavior, and the genetic diversity crashes. So this is cultural hitchhiking, and clearly it can work in my computer, but does it happen in the real world? That's a bit more controversial. The idea came from the precursors of the scenario shown here, where these are for all the cetacean species where we have a reasonably good estimate of nucleotide diversity of the control region of the mitochondrial genome. And that's plotted against latitudinal range. And you can see there's a generally good relationship, so the animals found in more diverse habitats have higher diversity. There are 5 clear, prominent exceptions. These are the killer whale, the sperm whale, the 2 pilot whale species, and the false killer whale. And these species have one important thing, at least for me, in common, in that they all have matrilineal social systems, in that daughters typically, usually always, stay with their mothers in the same group as their mothers when both are alive. So why is mitochondrial diversity so low in the matrilineal species? Well, a lot of people have thought about it and come up with several plausible reasons. And the most obvious one, which everyone thinks of when we've got low genetic diversity, is a bottleneck. There were very few animals back maybe during some ice age or during whaling. But why just the matrilineal whales? And if we look at nuclear diversity, there's no particular sign that these animals have reduced nuclear diversity, which is what you'd expect if there had been a bottleneck. The second common explanation is selection in the control region. Again, why just in the matrilineal whales? When the people doing genomics have looked at killer and sperm whales, they find reduced diversity throughout the mitochondrial genome, not just the control region. So a better explanation is a selective sweep in the mitochondrial genome. So somewhere in the mitochondrial genome, something really important has happened, and it's reduced diversity everywhere else. Again, why just in the matrilineal whales? And although there's sort of mixed evidence for this in this killer and sperm whales from the genomic studies. On the surface, perhaps the most obvious explanation is matrilineal demographics. And the idea here is because these animals live in matrilines, those are the units of population biology. So the effective population side is not the number of breeding females, it's the number of matrilines. I modeled this and showed that this actually can work, but it only works when you have unrealistically coordinated reproduction or mortality. The females have to give birth together, they have to die together. Now, in some of these species, they do strand together, but it's nowhere near enough to drive this. So that one doesn't work. And the last one is cultural hitchhiking. That's my favorite. I put up some references here, and this is just a selection. A lot of people have been writing and thinking about this. Well, now I'm going to summarize how I see this, first for the sperm whale. Sperm whales are matrilineal, and my student, Christine Conrad, produced a talk, which was on Tuesday, showing this very clearly. They have low mitochondrial DNA diversity. There's no evidence for selection in the control region. There might have been a selective sweeps. They have average nuclear diversity, which is evidence against bottlenecks. There are clear cultural groups which fit the pattern we would expect for cultural hitchhiking. These are the clans which have characteristic behavior and, as Marianne Marcoux has shown, differential reproductive success. So I think cultural hitchhiking, it isn't proved, but for me, it's the most parsimonious explanation for these guys. For killer whales, it's a bit more complicated. They are matrilineal, they have low mitochondrial DNA diversity, they have average nuclear diversity, but they have all these ecotypes. So things could be happening between ecotypes or within ecotypes. And among ecotype processes include the simple thing that if you subdivide a population, it tends to have lower genetic diversity. You could also have cultural hitchhiking on ecotypes, but that doesn't seem to work terribly well. I can't really see how mammal eaters could compete with fish eaters. The within ecotype processes are also possible. Now, you can get cultural hitchhiking within ecotypes, and Olga Filatova has suggested to me, and we've worked it up a little bit, the idea that the highly matrilineal resident ecotype in the North Pacific has considerably lower diversity than the somewhat less matrilineal transients, the mammal eaters. So this fits with the idea that cultural hitchhiking has been happening in the residents, but not as much or not in the transients. The new genomic work has pretty good evidence for both bottlenecks within ecotypes and selection within ecotypes. And especially when the ecotypes were founded, there were very few animals in each ecotype, which fits with the bottleneck idea. So if you put all this together, I'm going to bring together two quotes from very different places. The first from Lance Barrett Leonard, trying to give us a feel for the nature of the killer whale. Diet selectivity, xenophobia, and a strong dependence on culture. Second, a neat paper by Primo and Hublin, who looked at the potential causes of low genetic diversity in humans and suggested that in populations structured by culturally mediated migration, selection and potential bottlenecks can suppress neutral genetic diversity. So in killer whales, cultural mediated xenophobia sets up barriers, reducing genetic diversity. So I'm now going to move to the most obvious, or the most contemplated part of gene culture coevolution. Culture drives the evolution of functional genes within species. And I got an idea here when I was thinking about this 2 years ago. I'd seen Pittman and Durbin's great work on those pack ice killer whales beating their flukes in concert to drive the seals off the ice floes. And I noted that these animals had the biggest white eye patch. And I thought, well, maybe that's to do with coordinating their fluke beats. So I asked the genomics people, what do you think of this? And they said, basically, we think you're nuts. But we've got a really neat paper coming out. And they did. Oh, that's the eye patch. And Andy Foote and his colleagues' paper, Genome Culture Coevolution Promotes Rapid Divergence of Killer Whale Ecotypes. And they've shown that genes for adipose tissue development are enriched in polar ecotypes. The gene for skin development is selected in Antarctic ecotypes, and perhaps most dramatically, the genes associated with dietary variation and genes associated with the methionine cycle, which is related to diet, differ between fish-eating and mammal-eating ecotypes. And a key thing here is with the methionine cycle, they found differences both between the mammal eaters and fish eaters in the North Pacific and in the Antarctic. Different genes, but same kind of processes in the same area, which is similar to what we find for humans when we're looking for gene culture coevolution signals. Well, finally, I'm going to bring up the idea that this could happen species-wide. And the idea here is the genes drive the evolution of traits, the culture drives the evolution of genetic traits which promote the use of culture. So for things like extended juvenile period, in which the young can learn, it seems to make sense. I haven't found any evidence for it yet. There is a bit more here, the idea of menopause found only in humans, the short-fin pilot whale, killer whales, and false killer whales. And the idea here is that the post-reproductive life allows knowledge transmission to relatives. And Lauren Bent and her colleagues' work on leadership in killer whale does suggest that these older females have a role here, an important role. And finally, there's the cultural brain. The idea that because an animal is cultural, it promotes enlargement of the brain and cognitive enhancements that enhance social learning itself, that enhance the memory of cultural repertoire, and allow the animals to efficiently exploit culture. So finally, this is a table with the 5 kinds of gene culture coevolution that I've talked about, plus one other, gene geography, and where we think we have a little bit of evidence or good evidence in species. If you look through this table, who features heavily? Well, humans do. as we might expect. But who else features heavily? Cetaceans do. And in particular, those wonderful animals, the killer whales. So, in summary, I think the cultures of the whales and dolphins likely have had a role in behavioral plasticity, reducing selection, in the speciation process in killer whales, in reduced genetic diversity in the mitochondrial, in the matrilineal whales. There's been some selection for functional genes in killer whale ecotypes, probably driven by culture, and perhaps some species-wide functional genes have also been selected for. But I will admit there's much uncertainty about all of this, as in fact there is about gene culture coevolution in humans. But there's also much to come, and it's going to be a really exciting time over the next few years. Thank you very much. Oh, questions? Yes.
Thank you, Hal, for a very interesting presentation. So we do have time for questions. So if you'd like to go to the microphone, that'd be great. Thanks.
Great talk, Hal. Thank you. I guess my question is, do you think that what you see today is representative of what these cultures and these animals were prior to whaling, given that 700,000 sperm whales were removed from the ocean and actually not that many killer whales, probably relative to their abundance at the time and now?
That's a really nice question, and I think it's something we've thought about quite a lot. And in fact, one of the reasons we started thinking of it is some ideas you put out. And we think about this perhaps particularly with the baleen whales, which were reduced to such low numbers. And a parallel here is potentially with humans. So the Aboriginal humans with the simplest culture seem to be those from Tasmania. And the explanation is they've been cut off a small population for thousands of years, and they lose important cultural information. So if you get a bottleneck, so just a few animals, you lose both genetic information, as some gene copies are lost, but you also lose cultural information as information is lost. So you've suggested, and I think this is something we really need to think about, that some of the baleen whales used to use habitats which they don't anymore. And this includes some species which have come back to pretty good numbers, and they're still not seen in places where they used to be seen. Now, this could be partially due to oceanographic changes, but it could also be due to the fact that they've lost the knowledge that these are good places to exploit. Thank you very much, Damien.
Okay, thank you, Hal. That was excellent. So the fourth, last, but by no means least presenter for the plenary sessions is Nigel Hussey. So Nigel is an assistant professor in biological sciences at the University of Windsor in Canada, and he leads the Arctic program for the Global Ocean Tracking Network. His research focuses on the use of biotelemetry and chemical traces to study individual, community, and ecosystem level processes with the principal aim of providing data for effective conservation and management strategies. He does take on this ecosystem-wide approach to his research, so he has a lot of experience in a wide variety of marine species. Okay, so what can I tell you about Nigel that you may not know? So I did a similar exercise and found a colleague who was willing to speak to me. For him, science is not a job, it's a passion. And through this passion, he inspires countless students, co-workers, friends, and the general public to see what hard work and perseverance in science and research can accomplish. So I'd like to introduce you to Nigel Hussey, who's going to talk on advancing an ecosystem field approach to understand and manage aquatic predators.
Okay, thank you very much, Damien, and thank you very much for having me here today. Okay, so first, what we're going to do is do a quick recap of the plenary talk so far. So, we had Asha first, who spoke to us about blue whales off Sri Lanka, but in particular raised a very important point over the divide between the developed and the developing world in terms of research funding, and training and capacity. We then moved on to Scott, and Scott spoke to us about the Anthropocene and the impacts of humans on whale populations, and basically how we can define metrics to be able to understand changes in populations, especially given these populations are small sample sizes. And then we moved on to Hal Just, who's spoken to us about the evolution of culture. And now you have Hussey up here on the stage. So let's take all of these four plenary presenters together. And what we might start to see is there's an imposter here in the crowd. And that is, if this was America's Got Talent, Hussey might be getting a big X, because he appears to be studying the wrong taxa. But this is not quite true. I was speaking to my father a few days ago, and as you probably guessed by now, I'm from England, from the UK. My father reminded me of a story when I was much younger. I was born in Stoke-on-Trent, which is right in the center of the UK. It's about as far from the ocean as you can get in the UK. But as the picture demonstrates at the bottom, I had a clear love of the ocean. Whenever I had the opportunity, I was out there in the ocean. But one day, about 6 or 7 years old, my father discovered me behind our garage at the house, in a hole about shoulder height and about 10 feet long. And apparently, when he asked me what I was doing, I was digging a hole for the dolphin that I was going to keep in the back garden. So there was an association with marine mammals from a very young age. But on a more serious note, and how all good stories begin, is once upon a time, in a land far, far away, there were two magical animals. And these magical animals were the Greenland shark, as you now know, an animal supposedly the longest living vertebrate on the planet, and the narwhal, that I must admit, until over my 7 years in the Arctic working there, until about 3 years ago, I actually still didn't believe existed. But I was very fortunate to be invited to work with Department of Fisheries and Oceans. Now, for people from outside of Canada, Department of Fisheries and Oceans are one of the main organizations, particularly up in the Arctic region in my area, who do work for management of marine mammals. And these guys in the Arctic are particularly focused on managing stocks. Obviously, narwhals are important to the Inuits, the local people of the north. And DFO particularly have a role in understanding movements of these animals relative to stock structure. And I had some crazy ideas on associations between Greenland sharks and narwhal. This is really a talk for another day, or I can speak to you after this presentation. But I wanted to address questions over interactions between these two predators. And I was very fortunate to be invited to join the narwhal team, the DFO team. So we ventured off up to Greece Fjord. This was in 2014. Greece Fjord is the highest community in the Canadian Arctic, about 100 people. It's the true Wild West. And straight away you can imagine when you look at the photograph here is you start to realize the remoteness of this location to operate in the field. And so to be able to organize these camps, we fly in equipment, for example here, with twin otters, with polar continental shelf support. All of the equipment, all of the people have to be landed into sites. There's no runways. People are put on the ground in certain cases, and we actually build the runways there. And then we construct these camps. We operate on a very simple level, as you can see at the bottom, zodiacs with small engines to be able to work in these sort of unique conditions. And the uniting of the shark and the marine mammal team in the Arctic was actually very successful in 2014. As you can see here, I'm not constipated, but actually I'm pulling a long line by hand. This long line's in just under 200 meters depth of water. And the reason Bob is smiling at me at this very moment is there's actually three 3-meter long Greenland sharks on this line. What I'm not showing you is I actually only pulled the line for about 20 seconds and then I passed it back to Bob. It was just a photo op. Anyway, success of tagging these animals in the Arctic, as you can see here. And I need to point out in the bottom right, many of you might know is Jack Orr. And Jack Orr is a mentor, is an inspiration to me. And he's been the lead of the marine mammal, in particular the narwhal team in the Arctic, for over 30 years. But these camps in the Arctic over these 3 years really started a thought process. And this thought process started as Jack has now moved into retirement and a new person, Bob Hodgson, has moved in as the chief technician at DFO. And the thought process was that these camps are hugely complex to organize. They cost a huge amount of money, and the logistics of getting all the equipment into the camp is huge. Normally, these camps operate for 3 weeks. For decades, they were single species focused camps. But when you think about it, once you've actually got all the equipment in place and your camp is established, actually the cost to extend these types of camps becomes relatively minimal. And this was the thought process that Bob and I started over these 3 years in the Arctic was, okay, we've been focused on narwhal for how many decades in these camps? Suddenly we start to do Greenland sharks and narwhal, and we have huge success at understanding both of these predators. And also one of these years, note, we didn't get any narwhal. So the main success of the camp was actually the Greenland shark work. So then it started to push the idea of really we could extend these camps for relatively small amounts of money, and we could start to address what we should be doing, which is understanding these animals within their ecosystem. So this brings about the ecosystem approach. It's obvious. They've been telling us for decades that we should be studying animals within the ecosystem approach so we can generate adaptive management. But still, and I'm looking out at all of you now, I assume about 90% of you are still very focused on single species research, as I am. Most of my research program is still focused on single species. But it's obvious that we need to move into an ecosystem context. And just putting this onto a bigger scale, if we look at the direct value of coral reefs, seagrass, mangroves, and marine fisheries, it's about $6.9 trillion. This is not just fisheries. We need the ecosystems to support those fisheries. So the value is transferred across all of those realms. So from those initial camps and those conversations between Bob and I, the seed was sown. Little did I know where this little seed would take us. So 12 months ago, it started with endless meetings, hours and hours of speaking. We then sat and wrote endless grant proposals, trying to raise the funds to make this field work happen. And then finally, after a lot of praying, the money started to come in, and there was the reality that actually maybe we're going to make this happen. Of course, as we all know, there were lots of tears within this process. But finally, the project began. And I'm going to take you now to our ecosystem site in the high Arctic in Trombley Sound. And Trombley Sound is unique on many different levels. One of the reasons is it's a very important summering ground for narwhal. It's close to the community of pond inlets that rely on this particular animal for subsistence hunts. And it's also now part of the new protected area that's just been designated up in Lancaster Sound. So what did we want to do? Well, the key with this project, with our ecosystem approach, was to capture the temporal variability in the system. So what we really wanted to do was to arrive there as basically the ice was breaking up. So we were there at the very beginning of the season. And we wanted to study this system, this highly dynamic Arctic system, right the way through until ice reformed. So the camp team, the first core people arrived on the ground in 16th of July. As you can see here, plenty of ice still around. The core team set up the initial base camp and started organizing equipment. Just to give you some scale of equipment, we don't travel lightly when we travel up to the Arctic for field work, but to really give you scale in terms of what was achieved with this ecosystem project, I was based in Mary River, one of the largest mines in the Canadian Arctic, and I actually slung in with 6 helicopter swings, about 3,200 kilos of equipment into this camp. At the same time, Bob was based in the community of Pond Inlet, and he did 4 twin otter flights, bringing in about 3,700 kilos of gear. So you're looking here at about 7,000 kilos, 7 tons of equipment that was brought into a remote camp to make this work. Never mind, much of that equipment, the logistics of shipping that up from the south in the first place and actually getting this in the Arctic on time to make this all work. The camp grew very quickly. After the start, people arrived for leg 1. There was more camps for accommodation, more tents for accommodation, sorry. And by leg 3, we'd got an additional dry lab. So by now we had 2 dry labs and a wet lab. And by leg 3, we had the satellite seal tagging team arrive. All creature comforts were catered for in our camp. You can't really see me, but I'm here brushing my teeth with the biggest mirror in the world to look at in the morning. We had the perfect bathroom reeds in our bathroom facility. And we even had dog yoga. But really, I need to stress here, I am merely one voice talking to you because this ecosystem project was really only possible from a huge number of people, many of which are here in the audience today, and you can see here on the slide. So what did we do? What were we trying to do? Okay, well, we started at the base of the food web. I don't know anything really about the base of the food web. I don't think many people in camp did, but we went for it. We did plankton toes, vertical plankton toes, various depths, horizontal plankton toes, and all of this work the guys back in DFO were telling us is going to quantify species diversity, biomass, and we would also be able to validate putting in, for example, AZFPs, this equipment here, which we were going to put into the water not only for the 9-week period of sampling, but would be left in the water for the whole 12 months, and we will recover this next year. We then ramped up from the bottom of the food web up to secondary consumers. In this picture in particular, fight nets, but we used all different trap nets, box traps, jigging, every type of fishing gear you can imagine. We were trying to see what was out in the system. We moved up to tertiary consumers. We set various gill nets, various mesh sizes, targeting principally Arctic char, obviously a key species in the Arctic for the Inuit in terms of subsistence, and also for developing commercial fisheries into the future. And then all the members of the team, these poor guys, were not only catching all these animals out on the water basically 24 hours a day, but then they were back in the wet labs dissecting these animals, some real traditional hardcore science on trying to understand what these animals are feeding on, sampling these animals for chemical tracer analysis, such as stable isotopes, fatty acids, highly branched isoprenoid biomarker work. But the key again here was we had this temporal scale over 9 weeks to be able to sample animals and look at how stomach contents or feeding behavior changed, how condition of these animals changed over this time frame. We tagged various species within the system, and these really were sentinel species. So Arctic char, a sentinel species for the pelagic environment, to sculpin, a sentinel species for the benthic environment. And Arctic cod, a key species that links the base of the food chain, obviously, to the top predators, including narwhal. We shipped up piping into the Arctic, and in the field, we built our own baited remote underwater cameras to then go and sample mesopredators and predators in the system. And these were deployed every day throughout the 9-week period. We then sampled with long lines, fishing for Greenland sharks in the system. I think we only missed a few days over the 9-week period in terms of sampling. And then when we were catching animals, we attached a whole array of tags to these animals. Again, not just to address movements of the animal, but to address the animal within its ecosystem. So how the animal's interacting with its environment and how the animal's interacting with other animals. And how we did with this was through using archival tags that give us depth and temperature time series data, MRPATs, that actually allow us to reconstruct horizontal tracks for animals that don't surface. VMTs, these are mini receivers that we can put on the Greenland sharks, we can send them off, and then they can actually detect tags in other animals in the system. So then we can really start to look at Greenland sharks interacting with other species. CTDs with accelerometers to look at fine-scale behavior of these animals. And the first deployment of D-tags, hydrophones on Greenland sharks, to use them as bioprobes to monitor what are the ambient sound levels in the Arctic environment. And finally, we internally implanted acoustic tags in these animals, which is part of long-term tracking across the entire Arctic. Okay, so by this point, we're only at day 7. Day 7, the poor guys are absolutely knackered. They've been out there, they're doing everything possible. But now was time to move into the next phase of installing passive sampling equipment into our system to really drive forward the ecosystem approach. This started with servicing equipment. For example, here, these are receivers. These are basically the ears, the detect tags that we put internally inside fish. We also had hydrophones, such as SM3Ms, that were set up for recording marine mammal vocalizations, not just during the 9-week camp, but to be deployed for a whole 12-month period under ice. A key theme of our work in the Arctic, both departments of fisheries and oceans and myself, and a key focus of the work in Tremblay Sound ecosystem project was involvement with the Inuit and training, capacity building. So Asher, even in Canada, there's a problem and a divide between training capacity in the north and south of this country, never mind between the developing and the developed world. So here you can see Mapano worked with us the year before, and Mapano built all of the moorings that went into place in the pond inlet, or in Tremblay Sound. And then the Inuit as a team also deployed all of the equipment, as you can see here. So we built these gates of receivers and monitoring equipment that could basically track the movements of various species through the system. This included the receivers and marine mammal devices. So 30th of July, 2017 is one of those magical moments when you're in the Arctic. This was the day when the ice has disappeared and finally the narwhals entered the system. And on this particular night, about 2 o'clock in the morning, greater than 3,000 animals came in swimming past our camp. This is where we have the next game changer. At this point, we start to set fixed gill nets to do live capture of narwhals. So now the team switches into a 24 hours a day work program with the team rotating on shifts. Again, as with the sharks, whole series of tags, and we really addressed the technology that was being used previously to monitor narwhal, and we worked with the various companies and manufacturers to advance this. So we had backpack fast-lock GPS tags to give us very high-resolution location data. We had backpack CTD resolution SMRU tags, so the idea of animals as oceanographers in the system. We had backpack VMT-linked receivers. So VMTs are those mini receivers we were putting on the sharks that can detect fish. We linked these on the narwhal, so we could send the narwhal out and they could detect interactions with fish and sharks within the system. For the first time, we put mini-pack archivals and backpacked them on those backpack tags so we could get very high resolution 1-second dive data on the animals. We put accelerometers with CTDs, and we also put accelerometers with hydrophones, so again, recording the ambient environments within the Arctic. And then finally, in our sampling, was the ring seal team. Again, using satellite technology to look at horizontal and vertical movements. And then, as if we hadn't got enough challenges, we thought we'd take on one more challenge, which is the installation of moats with wildlife computers. Only the 3rd place in the world that these devices have been installed, so why not? Let's go and do it in the high Arctic and really put ourselves to the test. And what do these do? Well, these devices, basically, they bypass satellites, so we can receive all of the transmissions directly through these stations, which then automatically ramps up the amount of data that we're obtaining on these animals in such a logistically complex environment. In the end, it approximately doubled the amount of data that we were able to receive. And just to show you a level of coverage, on the left-hand side, we had one moat station that gave full coverage into Tremblay Sound at the bottom left, and right up into Eclipse Sound. And on the right-hand side, a second moat station at Bruce Head that looped right the way through Milne Inlet. And I'll talk a little bit more, but the priority in Milne Inlet is this is a major shipping route for transporting ore from the largest mine in the Arctic. So this was really going to give us very high-resolution data on narwhal movements relative to that shipping. Okay, so we're 6 weeks out of camp. I tried to spend 4 weeks of that in the pub. Unfortunately, I ended up back in the high Arctic. But just to give you a few sneak previews of what we were trying to achieve through this ecosystem approach led through Departments of Fisheries and Oceans. First, let's put the data in perspective. We've got 9 weeks, 10 terabytes of data. 10 terabytes of data is about 2 million IBM Model 350s in the 1960s. We're in an age of big data, but phenomenal the amount of data right across the ecosystem that was collected. At the base of the food web, a whole diverse array of species, from pelagic tunicates, arrowworms, sea angels, various species of copepods. And the guys back in the labs, I'm sure now, are very excited with the thought of the potential of new species that will be identified from such sampling that was conducted. And again, just keep in your mind this temporal transition, the breakup of ice to ice formation, and the ability to see these changes, the trends in these species. To me, one of the biggest surprises was in stomach content data. This idea of sampling animals on a regular basis. So this is stomach contents, and on the x-axis here, days. So sampling these animals throughout the camp, we saw very clear transitions in terms of what these sentinel species were eating. For both pelagic and benthic food webs, there was a clear shift from amphipod-dominated diets into larval fish-dominated diets. For the fish tagged in the system, about 180 fish were tagged. We did download some receivers. We had thousands of detections. But really, next year, when we go back in July and we recover these receivers, we'll have over 12 months of data to really understand the movements of these animals in the ecosystem relative to the predators. So, of course, to the coolest animal on the planet, you all know this, you just don't want to admit it. First, VMTs with accelerometers. So these are those mini receivers that can detect other animals in the system. What did we find? Well, actually, what we found is we got lots of detections of other sharks. These sharks were hanging around in the system. And what I'm not telling you is actually the correlation between the occurrence of narwhals and Greenland sharks was very high. For example, we fished at the start, just after ice breakup, no sharks, no sharks, no sharks, no sharks. On the BRUVs, the cameras, no sharks. As soon as the narwhal came into the system, the next day, sharks, 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 stemming from one shark up to 4 sharks. We could catch 4 sharks every 3 hours on 8 hooks. That catch per unit effort is through the roof. Why are these animals there together? Anyway, back to this. These interactions, the sharks staying into the system, they were detecting one another. And already I love this calculation, a jerk, which is a measure of activity, we basically showed that these animals were altering their fine-scale movement behaviors when they were interacting with one another. Yuki Watanabe, a colleague of mine from Japan, demonstrated a few years ago with Kit Kovacs here in the audience and Chris Liderson that Greenland sharks are the slowest swimming fish in the ocean. But are they? This is just a little snippet of an accelerometer showing you triaxial data on a Greenland shark. So those 3 colored lines are the 3 directions of movements. But let me draw your attention to this. This Greenland shark hit 8G in a rolling motion. So 8G, you're probably thinking, why 8G? Well, let's put 8G into perspective. This is a graph of the takeoff profile for the Discovery shuttle. It hits 3G. All Greenland sharks are 8G in a rolling motion. So they may swim slow, but they certainly roll very fast. Greenland sharks as bioprobes in the Arctic. This was, to me, one of the most amazing observations of using these animals. An animal that's moving very slowly through the Arctic environments, almost the perfect platform to put a hydrophone to observe the ambient sounds recorded. Kevin, do I hit this or is this you? Oh. One second. Kevin, is it me or you? So a key point here, these animals able to monitor the environment they're swimming around, but to me, incredible that clearly these Greenland sharks are swimming through areas where all the narwhals are within a relatively close range or close proximity to these animals. And then we could flip that on its head. So instead of monitoring the environment from a Greenland shark swimming through the narwhal, but actually the hydrophone on the head of the narwhal recording individual level interactions in the environment. Kevin. Focusing now a little bit more on narwhal, given we are at the Marine Mammal Conference. So a few last tidbits of data. This is 18 narwhals as of the 23rd of October, nearly 56,000 locations for these animals, showing connectivity between Tremblay Sound and Admiralty Inlet, which is the community of Arctic Bay. Huge implications in terms of understanding stock structure of these animals. If we take that and distill it down just to the journeys of 10 animals, this is just fast lock GPS data where we managed to get about 5-meter resolution in terms of location accuracy. This shows you the paths of those animals to date. And just honing in here, this is one animal, just to show you the difference between our Argos location data and our FastLot GPS data in terms of the precision of the tracks that we're able to generate. And this is huge in terms of trying to understand the fine-scale interactions of these animals within the ecosystem. For example, how they're interacting with their environment, but also how they're interacting with shipping and shipping traffic. In terms of diving behavior, this is one whale from 5 days. At the top, the temperature profile. In the middle, the diving behavior. And as you'd expect, as they move into deeper water, these animals are diving to deeper depths. But at the bottom, this chaos of these 3 colors, the orange, the blue, and the yellow, the accelerometer data, as these animals are diving in their diving process, really complex acceleration patterns taking place. We've already run, with the help of Alex here, sat near the front, and Fran, we've ran algorithms that are running through this particular data to break it down into different dive types. You can see here high acceleration U-shaped dives, and on the right, shallow, low acceleration V-shaped dives that tail on the way back up. You can see that the dive profile, the purple line, is overlaid with the high acceleration data, so showing very different acceleration profiles related to the dive profiles. And also, we can then overlay on top of that the number of clicks or the sounds recorded related to feeding from the AccuSounds. We can also, importantly, relate this back again to the environments and the ecosystem. So here's an unusual dive profile that we pulled out, a W-shaped dive, where you can see on the left-hand side as the animal's descending, the thermohalicline is at about 15 meters. Within a relatively small spatial scale, when the animal is ascending again, the thermohalicline is at 7 meters. So is the environment, these rapidly changing dynamics in the environment, modulating these movement dynamics? Here, diving behavior in groups. This data for a male and female, mature animals caught at the same time. The data on the left is hours post-tagging. Both animals actually identical in their dive profiles. But then, Luke, 5 days post-tagging, 500 kilometers away in Admiralty Inlet, and these animals are still showing complete identical overlapping dive profiles. So insights into the behavior of the animals. And then narwhals as bioprobes. Obviously, as oceanographers in the system, generating these very high resolution profiles. To date, we've got 2,000 CTD profiles with GPS locations from these animals in the system. And these data in the future will feed into an oceanographic model that's already been developed and they've had problems with, that's related to understanding ballast water related to the shipping in Milne Inlet. So a couple of last points so you can sit down, Damien. Don't worry, I'm not going to keep you standing there. I'd like to just reiterate a key point. The camps in the Arctic, our work is about training and capacity building. We had numerous Inuit involved in our camps, and this year was the first year that a full Inuit team tagged narwhal by themselves. Inuit were trained in surgical implantation of acoustic tags into all of the fish species. They were trained in building moorings, deploying equipment, recovering equipment, downloading data. So this is really working towards sustainability in the future. And as a final point, 2 minutes, as a final point is outreach. And really, we all know that the importance of our work is to translate this out to the broader public. And this has been a key focus of our work, and in particular, this idea of outreach at the ecosystem level. And so here, I'd just like to finish with one of those outreach videos that's been generated from the Tremblay Sound project by OceanWise, one of our collaborators.
This year was the first year, it was like the test bed of can we put a load of people on the ground and really go at trying to understand an Arctic ecosystem in very challenging conditions and a challenging environment. The number of people that you need on the ground to deal with large mammals, you can't just have 3 people in a field camp. You've got to have, up to about 14 or 15 people, including, obviously local guys from the communities here. It's logistically very expensive, so you invest a huge amount of money, a huge amount of effort setting up camps, moving equipment around the Arctic. You're always doubting if equipment's actually going to be there, and the actual logistics of the tents, all the food needed, the flights to get people up here, the challenges of moving people between base camp and back to Pond Inlet, long days and nights. I think there's been a lot of blood, sweat and tears by numerous people, involved, from DFO, academic institutions to the Inuit here in Pond Inlet, who were absolutely key on the project.
Good job. Good job. See, it was right in this area. True. See, you did good. Nice.
Tremblay Sound's very important on many different levels. It's very important for narwhal, which are a very valuable resource for the Inuit for subsistence, but also, obviously, you're an enigmatic, charismatic animal globally. An area in the Canadian Arctic where we've got this massive congregation of biomass during the summer, which clearly highlights it as an area that we need to protect moving into the future.
I'd like to finish with one thing. This is just recently I have a house near me that's been converted into a little pub. And I was sat there with a friend the week before I came here to give the plenary, and I just want to draw your eyes to the bottom of the table. Science is magic that works. And to me, that was this whole journey with the ecosystem project of Tremblay Sound. And with that, thank you very much for listening to me. Thank you to the conveners, and thank you to everyone involved.
Thank you, Nigel, for a very inspiring presentation. Unfortunately, there's no time for questions. But please do ask him at the coffee break, which is about to kick off. And here's your fishy, fishy, fish.
very quickly introduce literally a man that needs no introduction, because if you were here this morning, we know him as Dr. Alex Karamanlidis, Conservation Merit Award winner. Yes, do it again. So hopefully you saw his great speech this morning. I think he will touch on a lot of those points again about the work that they do. And I guess I will only take this opportunity to say I've been working with Alex for quite a long time now for sister species. He's my brother on another sister species. Sharing information, sharing the struggles that we have. So we've had a couple of opportunities where we've spoken together on the stage or to different audiences, and it is always an intense and great pleasure to be able to share a stage with you on our continued fight for conservation. So without further ado, Dr. Alex talking about their work with the Mediterranean monk seal. Other presentation. Yep. Okay, thank you, Alex. You don't want me to really sit up here, do you? I'm going to sit down here.
Yeah, whatever you want. So let me figure out first how this works. Okay, thanks Charles for the introduction. I'm happy that the, that the blood is flowing through my brain and I can actually understand what I'm talking about now. So let's give it a start. I'll talk a little bit about the Mediterranean monk seal. And let me see if this is going to work. Okay, so a little bit about the information on the taxonomy. Some of you might have heard that in a couple of years ago that the genus was split in two. So the Mediterranean monk seal is the only species of this genus. We have the Caribbean monk seal and the Hawaiian monk seal forming now a separate genus, the Neomonachus. We will focus now on the status of the Mediterranean monk seal. A lot has been said about the Mediterranean monk seal in the past 30, 40 years. It was destined to be one of the first marine mammals to go extinct. This is not the case, as we all know. It's considered now to be endangered according to the IUCN Red Data List. And as you can see on the map behind me, and the pointer should be working like this. Okay. We have nowadays 3 populations remaining. There is one in Madeira, up there, with a population of approximately 50 individuals. There's another small population, geographically small population in Cabo Blanco, with approximately 250 individuals. And there's a larger population in the Eastern Mediterranean, approximately 350 individuals. So the total population is below 700 individuals. What's very interesting to see is that the estimated area of occupancy is almost 315,000 kilometers, of which more than 90% of this is located in the Eastern Mediterranean. In my presentation today, I will focus on the conservation efforts that are being done for Mediterranean monk seals in Greece. And why is Greece so important for the species? Well, this is due to one main characteristic. The coastline of Greece is more than 15,000 kilometers, which makes it the 13th longest coastline in the world. But if you take that compared to the country size, we are one of the top 2 or 3 countries in the world. We have more than 4,500 islands, which makes it potentially an ideal habitat for the species to survive. Until now, we have found key populations in several islands, on the western side of the country, in the Ionian Islands, and in the eastern part of the country, in the Aegean Islands. The total population in the country is estimated to number less than 350 individuals. Again, getting back to the important fact here, you have to consider that, that more than 60% of the area of occupancy is located in Greece. Which is not to deter from the point of interest that the species is strictly protected since 1981, and it's on the endangered data book. But unfortunately, human-related cause mortality, like deliberate killing, accidental entanglement in fishing gear, and habitat destruction have played an important role in reducing population numbers over the past 50 to 60 years. I would like to focus now on the conservation efforts and research efforts that have been done by the Hellenic Society for the study and protection of the monk seal. We're also known as MOM. We perform activities in 5 principal areas throughout the country. The first key activity is environmental education. So we try to educate the next generation of environmental friendly and sensitive citizens. How do we do that? We do that with, for instance, 10 educational suitcases that travel throughout the country, where teachers in schools can use them to teach the kids what monkshood conservation is about. We have a summer school in one of the primary areas of distribution of the species at Alexandros. We have a special website for kids, where the kids can go with their teachers and learn more about Mediterranean monk seals. And we regularly do presentations at schools. So we estimate that more than 700,000 kids have been educated about the Mediterranean monk seal. Public awareness. Well, we try to raise public awareness both nationally and internationally for the species. We produce visual material, including posts on websites, radio spots, press releases, newsletters. We're very active on the social media nowadays. As I said, we have the kids' website, but also our own website. And we operate information centers during the summer when all the people are visiting the Greek islands for holidays. They can learn a little bit about the Mediterranean monk seal and why it's so critically endangered. We have a volunteer program that operates both in the rehabilitation center that we have and also in the summer information centers. Rescue and rehabilitation is one of the main activities that MOM is performing. So we have the Hellenic Monk Seal Rehabilitation Program that was established in 1990 and has been operating continuously since then. It's carried out in collaboration with the Aristotle University of Thessaloniki. And until now, we have been able to rehabilitate 29 pups, of which 17 were successfully released back into the wild. At the moment, we have 2 pups in our rehabilitation center, so whoever's lucky and visits Greece might be able to pass by and have a look at them. Unfortunately, our rehabilitation facilities are only destined to receive only small pups. We treat adults and sub-adults all in the field, and until now we have been able to treat 36 of these animals and release them back into the wild. One of our key activities is in situ habitat protection, which in practice means that we help the Greek state establish marine protected areas, and wherever we can, we help them in patrolling and monitoring these areas. So, for instance, in the northern Sporades, the islands where one of the biggest national parks in Greece has been set up, we performed for several years the guarding efforts. We participate in the management efforts of other protected areas, and as I said before, we help the Greek state provide information to it to establish new protected areas, both for the species but for the whole ecosystem in the country, the whole marine ecosystem in the country. Of course, we're very active in policy making. As I said in my presentation today in the morning, that we have a Byzantine-like bureaucracy which makes conservation efforts a little bit difficult. So we have to be very active in persuading with scientific data the Greek state to move on with monk seal conservation. We have participated and set up the action plan for the Mediterranean monk seal in our country. We participate in the National Stranding Network that is operating throughout the entire country and provides information about stranded marine mammals in general. We participate in the IUCN Specialist Group. We were part of the team that reevaluated the Mediterranean monk seal for that red data book, and we're preparing now a new evaluation of the species on a subpopulation level. One key activity which is very close to my heart is scientific research. So one must consider that when we started 30 years ago, nobody knew anything reliable about Mediterranean monk seals in Greece. We have, over the past 30 years, tried to increase our knowledge about the species, and we do that through the establishment of a rescue and information network, in situ monitoring of important subpopulations, and ex situ monitoring of the monk seal population in Greece. We have set up several research partnerships with research institutes all over the world, and our work until now has resulted in 38 publications. For those of you who are interested a little bit more, you can visit our website and have a look at them. At this point in time, MOM has established itself as the national authority for the collection and evaluation of monk seal sightings in the country, and we are an accredited research institute according to the CITES international regulations. One of the main tools we have for studying the species is the Hellenic Rescue and Information Network. It's kind of a citizen science project where people all over the country provide information about monk seal sightings. It was established in 1991 and has been operating since then. And below that, you can find some of the results up until now. So we have a network of more than 2,500 members, primarily on the coastal part of the country, who provide us information of regarding monk seal sightings. This has resulted to approximately 6,000 of these sightings, more than 1,300 photos and videos. But more importantly, it has enabled us to establish our monk seal ID catalog. So we have a catalog of photographs with animals from throughout the country. And very important for what you will see later on is the establishment of a monk seal sample bank, which provides the material for the ex situ conservation efforts. More importantly, of course, the whole system provides us the opportunity to operate an emergency team, which means that whenever we receive a phone call regarding an injured, an orphan, or a sick pup, or an adult individual throughout the country, we are able to operate, to travel. You must imagine that this is not, of course, an easy task to do. With more than 4,500 islands, traveling to one of these is not one of the easiest things. Moving on to in situ monitoring. So we have established now a network of automatic monitoring systems. As you can see on the map, we have 2, 4, 6, 8 areas throughout the country where we have set up automatic monitoring systems, cameras in the caves to monitor the population. This has resulted into more than 500,000 of photographs, which unfortunately we not have had the time to see them all, but we'll get up to that there. Ex situ monitoring. As I said, the Rescue and Information Network provides us the opportunity to go out and collect samples from dead individuals when they are found, or from live individuals when they're going into rehabilitation. So we have a big sample bank of more than 4,000 samples, which we can use for different studies. I provide three examples of these. The first one is a trace element analysis that showed that pollution plays no role in the threat status of the species at this time in Greece. We did a stable isotope analysis a couple of years back using samples from stranded animals and found that the species is a coastal feeder, eating mainly octopus, no differences between males and females. And one of the more innovative studies that we carried out recently with the support of the Marine Mammal Commission was the mtDNA analysis. As you can see on the graph in the middle. Yeah. So we analyzed samples of Mediterranean monk seals from the eastern part of the Mediterranean, and then samples from Madeira and Cabo Blanco in Mauritania. And we found out that the two populations have been separated, and you can see that through the fact that monk seals in the eastern Mediterranean have 1, 2, 3, 4 haplotypes, and they do not share any haplotype with monk seals in the Atlantic. We are planning to extend this study now on microsatellites, so we will have a little bit more clearance about this population structure. But considering the distance of these two populations between them, the Atlantic and the Mediterranean, we don't expect very much to change. So where do we stand now in time here with the conservation of the species? Well, at the moment we have set up a new project that we call Mapping for Conservation, and it's an effort to identify all the pupping sites of the species in the country. We have changed a little bit our conservation approach recently and are trying to find these areas because we believe that these are the most important places that have to be protected and to be set up as protected areas for the conservation of the species in the country. You can see the timeline. In the past 3 years, we have managed to monitor several areas. Next year, we will go down to Crete, the island down here, to see what's going on with the pupping habitat over there. And pending funding then in 2018, we will try to complete the entire coastline of the 15,000 kilometers in our country. At the same time, we have set up a new project. It has become evident that Mediterranean monk seals are expanding. They're moving out of Greece and going to the other areas. We have had opportunistic sightings in Cyprus, Croatia, Libya, and all the countries that are around Greece. So the next move is to establish a monitoring framework throughout the countries in the Adriatic. So in the next years, starting from 2018, we will be setting up a monitoring project for Mediterranean monk seals in Albania, Montenegro, and Croatia. Finally, the conservation priorities for monk seals in Greece in the next years to come. So we will try to enhance and improve the rescue and information network. The participation of the locals is very important to us, so we will try to expand that. We will try to improve the rehabilitation infrastructure. So we have made new rehabilitation facilities down in Athens where we can host 2 to 4 pups. We hope to find funding to increase that and perhaps be able to treat also adult animals. We will try to complete the mapping of the important pupping sites in the country. Of course, we will continue to work on the environmental awareness and education campaign that we have been carrying out for all these years. And of course, that's one of our main aims, because it's a big problem still in Greece, to improve the management of the already existing protected areas. This is all nice on slides, but I think it's even better if we can see it on the video. Kevin? you. So I guess before closing, I would like to mention that the takeaway message here is that obviously the Mediterranean monk seal is not out of the dark. With fewer than 700 individuals, the species might be listed as endangered, but we all understand that it's really critically endangered. But it's more likely that in the next few years, considering where we are now, you'll hear more positive news than negative news. And we hope that it will continue like that. Thank you very much. So speaking of good news, I think we can continue with the good news. For the past years, almost more than a decade now, we've been working with Charles, and as I said in my presentation in the morning, we have been inspired by the Hawaiian monk seal research project. The news coming from there is also very, very positive, and without further ado, one of the most eloquent talkers out there, Dr. Littman.
Okay, and for those of you that had questions for Alex, we are gonna, hopefully we'll power through this relatively quickly, and then we will answer questions collectively. Okay, so I'm gonna go about this a little bit differently than Alex. It's always interesting watching a Mediterranean monk seal talk. There's so many parallels in the issues we're facing and the species we work on, but there's also very stark differences about what methods work and what methods don't. And, you know, Mediterranean monk seals are such a great example of grassroots efforts to conserve a species, and Hawaiian monk seals are the exact opposite. So intensely government-driven and now evolving towards sort of greater collaborations, and I'll touch on that in just a moment. I'm going to, there's a lot of Hawaiian monk seal science that was shared at the meeting today, or this week, including another talk tomorrow morning, Jessie Lopez talking about contaminants. Bolander, sorry, she's married, I keep forgetting. And so catch her talk. I'm going to focus more on the arc of the history of monk seals, their resilience through time, and where we're currently at with the species and what we're thinking for the future, with the intent of leaving everybody inspired and hopeful for whatever your conservation projects might be. So we're going to jump back millions of years. I promise this will be very brief, and I'll only do it with 3 images. Sometime, we don't know when, monk seals found their way out of the Caribbean across what would eventually grow into Panama once it emerged from the ocean, and found this remote archipelago in the middle of the Pacific and made a home there. And we don't know if they were there for thousands of years or tens of thousands or millions of years, but they lived their lives, thrived, were unmolested until, as often the case, humans arrived. And based on archaeological evidence and looking at modern Hawaiian culture, their stories and chants, it is probably very likely that monk seals were extirpated in the main Hawaiian Islands. These are the high islands that many of you would be familiar with, shortly after humans arrived. And so it was the remote northwestern Hawaiian Islands, these small islets in the northwest of the archipelago, that was their place of refuge. And once again, they did pretty well up there, we think, until Westerners came and ruined it for them. And so these islands were rich with birds, so there was guano mining that happened there. During the last death throes of the global sealing effort, they started targeting monk seals. They didn't have particularly great fur. Oil probably wasn't the best. And decimated populations and caused local extirpations and probably brought the species to the brink of extinction. But they're resilient, they recovered. And then the next phase of sort of perturbation up in the northwestern occurred, and that was when the military came in and started modifying some of the habitat and building infrastructure. And in those places where there were humans and seals, we saw population declines. And once those the military presence left, we saw them rebound. So the modern era of monk seal conservation starts in the 1950s. Don't worry, the picture will come in a minute. In the 1950s, when some wildlife biologists up there did beach counts to do a general census of the population and counted a number and then largely left monk seals alone. And there wasn't much thought given to them, and there was a few research projects here and there. But in the 1970s, people started doing those same beach counts again and had realized there had been an incredibly dramatic decline between those two data points. And so very quickly, monk seals were listed under the Endangered Species Act as endangered and protected under the Marine Mammal Protection Act, and so they became NOAA Fisheries' responsibility. So in the 1980s, we started setting up field camps. We had phenomenal fashion sense. While they were also brilliant dressers, they were brilliant scientists. And we didn't know anything, or we didn't know very much about the species at the time, and we didn't really understand the threats. So I cannot thank the early forefathers and foremothers of this program for setting up such a high-quality population monitoring system that allows us to know so much about the demographics, the trends in the population, and feeds every recovery activity that we do. And it was that foresight that gave us the foundation of the incredible high-quality science that we have to work with today. So they set up these systematic field camps in the northwestern Hawaiian Islands, and over the years they grew and expanded and changed their mission. So it's funny, she said it's just population assessment and some conservation or recovery efforts. It's an incredible amount of work that our field teams do every summer, and again, they're part of the lifeblood of our program. I've got some examples in this next slide, which I'll narrate, about some of the things. This is just a random assortment of activities that they do, but they're incredibly important for the conservation of the species. So you can hit play, Kevin. Thank you. So marine debris is a huge problem. We get to get all of the debris that's collected around the entire North Pacific dumped onto Hawaii. Monk seals are very naturally curious, or they'll be feeding on the nets or trying to sleep on the nets, and they grow entangled. So we have a lot of disentangling that we need to do. And as Jessica mentioned in the video before that, pulling marine debris that comes on shore. We also have a very large-scale marine debris program, which is shrinking in NOAA, but has been important for helping the species. Another tool that we use is translocation, and this can be on a variety of scales, and it's very simply moving animals from an area of low survival to high survival. Here's a Galapagos shark, unique predatory behavior of beaching itself or taking young pups in very shallow waters. Kills about 25% of our cohort every year, and is the #1 threat for monk seals in French frigate shoals. One location, it's the only place it happens. We do have a shark removal program there to get these sharks that do this behavior, but we can also take these pups once they wean. Sometimes we will force wean them if there seems to be a high degree of danger, and just move them a mile away or a couple of kilometers away, and they do fine with no shark predation. But we can also do larger scale translocations, and this is a good example of how we can leverage the quality of our data, taking an animal from one subpopulation to another, where we can tell from the data that there's a good chance they're going to have a greater chance of survival. Often we focus on young animals, almost always young females, to salvage that reproductive potential, move them to the beach. The young weaned pups are incredibly docile and easy to move. And then we will monitor them to make sure that the activities that we're doing are fulfilling our goals and that we're being as successful as we need to. But probably one of the most important recent developments is our work that we're able to do rehabbing juvenile seals now and recently weaned pups. Historically, there's been chronically poor juvenile survival, with many of them not being able to find sufficient resources. So when we leave every year from our field camps, we would know we were leaving behind several animals that were going to die. The Marine Mammal Center in Sausalito, California came to our aid. They built a multi-million-dollar monk seal hospital in Kona on the Big Island in Hawaii. And it is a perfect balance of public partnerships. We go up with our vast resources in our large ships. We pick up the patients, bring them down to the hospital. They fatten them up. We bring them back home and release them back into the wild. And it is an incredible additional tool that we just only recently added in the last few years. We talked about the military's presence up there. There's a lot of infrastructure that was built. We do have a problem that infrastructure is breaking down now. So our teams not only look out for monk seals, but they protect turtles, birds, and of course monk seals from getting entrapped and entangled. Monk seals have an innate gift of getting themselves into terrible positions. This last one is kind of funny, but it's a warning that you always have to be on your toes for the next threat. And it speaks to something that Hal Whitehead spoke about this morning. So our team was walking down the beach, saw something sticking out of this seal's nose, discovered it was an eel, called us and said, do we have a protocol for removing eels from noses? Which we didn't. We do now. And so they went and captured it and started to extract this eel. I'm not sure if this is life-threatening or not, but it's fascinating. It's like a magic trick. Still going. So it was roughly a little less than a meter. The eel did not make it, but the seal seems to be fine. The fascinating thing, if you saw Hal's presentation this morning about culture and learning, in 30 years of monitoring the population, we had never seen this. No seal had ever had an eel in its nose. And this was at Lisiansky Island this past field season. So 1 year later, they had 2 seals, juveniles or weaned pups, that had eels in their nose at the exact same location. So I don't know if this is a hip new thing that kids are doing and it's going to continue to pass on, but we might be able to work with Hal to talk about cultural transmission. Those efforts have been going on for years and very small scale, opportunistic. We've moved into like translocation and rehabilitation, which are occurring at a much higher level. But while we were doing all that and focusing in the northwestern Hawaiian Islands, remember, monk seals had been extirpated in the main Hawaiian Islands. Monk seals proved impossible, or did the impossible, or what we thought couldn't be possible. They had demonstrated to us that they were very susceptible to human disturbance, so we never expected that they would take root in the main Hawaiian Islands again. And sure enough, we turned around and they had. So we're currently at a population of 300 seals. For a very long time, the population was growing very quickly. We didn't need to do anything to conserve them, but we were suddenly confronted with having to build a culture of coexistence with the species. So we went from science, conservation, dealing with large ecosystem types of things to interacting with people. And we initially were not very good at that. So we had problems with close interactions. Obviously, where there's fisheries and seals, you have interactions. So we do a lot of surgeries and emergency interventions. And then where marine mammals occur, particularly seals and people, there is conflict that often ends up in intentional killing of seals. This was a real blow to us. People that have been working in the northwestern Hawaiian Islands for years trying to conserve the species, and then finally there's this success story, and not everyone shared that perception. We had lots of public meetings, and I'm only going to go in the very shallowest of this process, but many public meetings where generally it was the local communities, particularly fishermen, that were very happy to share some very strong ideas about monk seals. And a lot of it was a lack of good information. But the two I'll focus on is monk seals eat up to 600 pounds of fish every day. He was an American fisherman, so he doesn't use metric. But that's the size of kind of the largest monk seal that you're going to see. And so we all know as scientists that that's not possible. But the one at the bottom is the one that really touches me, and it's this concern that these new, large, fat animals that are feeding on marine resources could potentially impact people's ability to bring food home to their families or their livelihoods, or potentially impact cultural practices. So we decided that perhaps all of our good science wasn't reaching the people that it needed to. And if you think about it, we're going to often Hawaiian communities of long-term fishermen. I'm a white federal biologist walking up to them and being like, look, I put satellite tags on them, I clean their poop, I could tell you everything you need to know about what they do. They just look at you like you're very strange. So we realized we weren't being transparent enough. So we decided collectively to blow up the way we do our science and try to be much more inclusive with the communities we're working with. So if our SCAD analysis and our satellite tracking wasn't touching them, we wanted to use a different tool, and that's the Critter Cam, to work closely with the community. So we worked with the fishermen to design the study. When we would go out, we would try to bring people to work with us. We were worried that we couldn't get anyone to watch all 120 hours of video footage we might collect, so we worked with classrooms that as soon as we collected the data, we sent it out to classrooms so no one could say we manipulated it, so before we even saw what was possibly on the tape. And then we would go back out and share this information, and we would hope that those students would support us. These children in those communities would say, we watched it, they're telling the truth. But the most powerful thing about this tool is it's an equalizer, right? So the fisherman's actually the expert, because I don't know what most of these fish are. I just know if a monk seal is eating it or not. And so it has, I think, been one of our most powerful tools for, I think, both letting people understand and appreciate monk seals, but also by creating a stronger relationship of trust, because we stepped out of our federal role and tried to be part of the community in the way that we did our science. And this is just a shot. We've worked with classrooms. We're working on a seal that's sedated, and then walking them past very quickly so that they can see what we were doing and be connected with it. And this is a Hawaiian immersion school. The teacher was crying. She thought it was amazing. A couple of these went on to do a science project, and I went back to visit them one day, and they were telling me how they were yelling at their community for saying stupid things about monk seals. I tempered that a little bit, but it is, you see this change instantly. It's amazing. So the last big thing that has happened is that our community has grown. So, Alex's grassroots to a broader partnership. We were a single government entity to an array of partners, and it has changed the world that we work in, and it's incredible. And it's, I think, particularly possible now because we are working in the main Hawaiian Islands. So where are we with the state of the monk seal population, and why should we be hopeful? The population's 1,400 animals. It's been growing in the last 3 years. 30%, and this is for all of you guys doing conservation, 30% of the seals alive today is because of R, and it's the collective R. It's all the scientists, all the managers, all the community that have worked to do it. Little things over a long period of time can make a huge difference for the species that you're working on. Public perceptions are changing. People are more accepting of monk seals. A very brief example, this is Kaimana. She was born in Waikiki Beach. We thought it was going to be a disaster. This is our tourist nexus in Honolulu. Millions of people every day. Had to shut down part of the beach. She was live streamed around the world. Everybody loves Kaimana now. It inspired a beer, which I have one here now. So if you can come and find me and tell me, the first person to do it, what Kaimana's mother's name is, I will give you this beer, fresh from Hawaii. But this is a local company that has made a beer, and the money and the proceeds go back to the volunteer community that was taking care of Kaimana. So people are starting to step up and do things that we would have had to do in the past. Creative ways to engage people, working with artists. We had this woman, April Surgent, embedded in our camps last year for 4 months. She did everything our scientists and our conservation people did, and then distilled that into artwork. So if you're back in Seattle or near until November 4th, check out her work or look at her for this online at the Traver Gallery website. So great things are happening. I want to give us a chance to answer questions. Sorry, Alex. But there are things to be concerned about. Disease prevention. We have vaccination program for several diseases, but now we're in a battle over feral cats and toxoplasmosis, things we never thought we would have to deal with. So there's always something on the horizon that you'll never quite expect you'll need to get engaged on. And then, of course, climate change. And how do we build the resiliency for the species to deal with the threats they're going to face in the future? These are the things we're starting to work on now. And so with that, I do want to acknowledge there's several people in my program that are here. They're the ones that I get to talk about this. They're the ones that do this. Legacy of scientists and managers that came before us, the massive and wonderful partnerships we have, including our partners overseas that help us find our way in conservation. So for all of you conservationists out there, keep fighting. You will absolutely make a difference. So thank you. Technically, we are done, so you may go, but if you'd like to ask questions, we'll stick around until we get kicked out. Does that sound right? Yeah.
Is this working? Yeah. So clearly amazing work you've been doing in Hawaii and also in Greece, and I'm just wondering how some of it's carrying over to the group in Mauritania and the other population in the Mediterranean.
Okay, so the situation in Mauritania is very complex. It's a population that suffered a severe mass die-off in 1997, where almost 2/3 of the population were wiped out by that die-off. Since then, the news has been only encouraging. The population has been increasing. I think the last year they had a record pup birth of 80 pups. The only problem is that the population there is very constrained. It's constrained to a 3 to 4 kilometer stretch, and that's our main concern. So I think that our colleagues from Spain who work in Mauritania will try to initiate efforts to expand this population geographically.
Yeah, Alex, I was interested in the genetic differences you've talked about between the Mediterranean animals and the Atlantic animals. Or is the intention now to manage the two groups separately as though they were separate species?
Yes. The two populations are managed separately, and we're moving now on to a separate subpopulation evaluation for the IUCN Pinniped Specialist group, because although the species in overall has been listed as endangered, the subpopulations on their own are most likely critically endangered. And we would like to draw attention to that fact that all these small populations are still critically endangered. So yes, they're managed separately.
Okay, and I was just wondering about the translocation work that these guys do in Hawaii and that you do in Greece. That video showed you releasing an animal, had been rehabilitated and was being put back, but was being put back in the same area where it had been recovered?
Well, first of all, I wouldn't go and use the term translocation. So we find the pups perhaps on a neighboring island, and we always release them in the center of a protected area. We know from genetics that most likely all the animals in the Aegean have the same haplotype. In the Ionian, it's a little bit different, but we do not mix the populations. We try to release animals in the center of a marine protected area so that we have a little bit of a more protected status. But as all animals, they just wander off, and then after that, they're on their own. But we try to release them in areas where we can have some level of protection for them.
Hi, Charles. Thanks for your talk, both of you actually, for your talks. I've been a long admirer of the monk seal program in Hawaii. And so I'm sorry to everybody, I don't actually have a question to ask about monk seals, but I have an answer to your question, Charles. And I was in a race with another person here to get to you first, but in the spirit of Ohana, we are going to share your prize if you're willing. And we believe that her mom's name is Rocky.
That is correct, and I'll give you both a beer.
Thank you very much.
While that happens, I'll just ask you a question. Well, first of all, thank you both for fantastic talks and for the hopeful message, which is sorely needed nowadays, I think. Alex, I'm curious, you mentioned that there's still intentional killing of monk seals, Mediterranean monk seals by fishermen. How intractable do you think this problem is? What are their perceptions? This is kind of a big question. Is it mostly commercial or mostly recreational fishermen? Octopus fishermen? Can you kind of generally frame what you really see as the challenges in sort of communicating with the fishing community? And are we speaking the same language, and can we?
Okay, so intentional killing of Mediterranean monk seals is mainly by commercial, small-scale coastal fishermen. So the species is a coastal feeder. It does not interact, or it interacts very little with open fisheries, so far away from shore. The coastal fishermen, of course, are distressed about the damage that the species does onto their nets and onto their catch. Nowadays, of course, and with the financial crisis, but also with the public awareness, the younger generation of fishermen is more aware about the difficulties for the Mediterranean monk seal, but also it has become harder for them to earn a living. So they, from what we understand, spend less time trying to hunt and kill the Mediterranean monk seal. That does not mean that the problems are over. Yeah, of course, in a country that's stricken by a financial crisis, when you lose your whole fish catch due to a monk seal, that's not very, very nice. I can understand that people going out and spending 2 days out and fishing and they come back with nothing are totally distressed about that. That's why intentional killing still persists, but it's definitely, and we do not have any hard date on that, but it's definitely less than it used to be, without saying and implying that it's non-existent.
Thank you very much for your inspiring and exciting talks. I've got a question about the Hawaiian monk seal. Do you have any information about the prevalences of toxoplasma, and is there a kind of eradication program or any program to prevent cats from having contact with seals?
So we're currently waiting on some results to have a much better idea about the prevalence. So, and these numbers always seem very small when we talk about this, and I'm trying to remember the exact, I think it's 8 confirmed deaths from toxo, or toxo and another co-infection. We think that there's probably animals, we certainly know that there are animals carrying it, and we are, again, results are coming out to get the total prevalence. We think that there's mortalities that are occurring that we're not detecting. And there is some very early concerns. You know, if you're a healthy animal or a healthy person, you're not going to, you know, the disease won't come to fruition and suffer the recourse of that. There is some little concern, and we're going to wait till we have more numbers, but that perhaps pregnant females may be disproportionately impacted. We have had fetuses aborted, but we're also concerned about the mother's welfare as well. Are there plans for eradication? As you can imagine, dealing with feral cats is incredibly sensitive, particularly in the United States. We look enviously at other countries that are more proactive in dealing and managing with the situation. There are millions of feral cats in Hawaii. In very difficult habitat with lots of endangered species, shorebird or seabird, ground nesting seabirds and stuff. We have a group that we're working with, trying to find solutions, trying to have very difficult conversations, but as currently it is primarily trap, neuter, release, and not very many legislative tools to use to try to deal with this. So, but hopefully in the future there'll be some further developments.
Hi. Thank you both for your talks and for the excellent work that you're doing. I have a question for Alex. There's been a lot of concern recently in Greece with the oil spill. So my question is, have there been any records already of monk seals being impacted or any concerns for the recent future? Direct or indirect?
Well, yeah, of course, there's concern. The Aegean Sea is a very closed sea, so you can imagine that an oil spill somewhere in the middle would be a huge disaster for the entire population living there. It did not affect, from what we know until now, the population now, because the oil spill was in the center of Athens. Unfortunately, there is no mitigation plan from the central government authorities to mitigate such a thing if it happens somewhere in the central of the Aegean. We have known for a while, of course, that oil residue that comes from ships and traveling ships throughout Greece is a littering factor within the caves. So you often go into the caves and find tar and oil residue overall. Doesn't seem to affect the species, but if we would be talking about a really big oil spill in the center of the main archipelagos in Greece, yeah, that would be a main problem, and we just hope that it doesn't happen. I think we can wrap it up for today. Thank you for your interest, and good luck. Thank you. Hey man, it was only